Welcome to This Explainer. I was digging through some recent research, and we are literally on the verge of a massive conceptual shift in how we might deliver complex biologic drugs. We're talking about completely rethinking how medicine gets into the human body. Instead of relying on external bags of fluid constantly dripping into a vein, what if we actually turned the body itself into the manufacturer? I mean, it sounds kind of like science fiction, right? But seriously, what if your body really could manufacture its own cancer medication? Well, thanks to a groundbreaking new study outlining two incredibly cool AAV-mediated platforms, aptly named TransJoin and TransSkip, we're looking at a medical horizon that might just eliminate the need for those exhausting, continuous drug infusions altogether. Okay, let's dive right into this. 2 hours. That short little window represents something incredibly frustrating in modern oncology. It's the grueling 2-hour half-life of a clinical cancer drug called blinatumomab. It's a highly effective bispecific antibody used for leukemia. But because it breaks down so insanely fast, your body essentially clears it in just 2 hours. And honestly, that rapid clearance places a tremendously heavy burden on patients. Because the body clears it so quickly, patients are literally tethered to continuous IV infusion pumps for weeks on end. Think about what that actually means. Repeated, exhausting hospital visits, dealing with pharmacological peaks and troughs where the drug levels dangerously fluctuate, and, understandably, a massive drop in patient compliance. It's just a restrictive, exhausting way to try and heal. So this brings us to an absolutely incredible paradigm shift. Normally, when we think of traditional gene therapy, we think of it as a tool to replace defective or broken genes, right? Kind of like fixing a permanent typo in your DNA. But this new AAV drug delivery method, it does something entirely different. It uses that exact same viral vector technology to install what is essentially a biological 3D printer inside the patient. It continuously manufactures therapeutic biologic drugs. We aren't fixing a broken gene here. We are literally installing a microscopic pharmacy. Enter TransJoin. This is an AAV vector engineered to continuously secrete therapeutic bispecific antibodies after just a single one-time intravenous injection. Think of TransJoin as the ultimate solution to the IV drip. By injecting this vector, you create a permanent in vivo factory that perfectly mimics the effects of that leukemia drug blinatumomab. It's constantly producing the medicine right there in your bloodstream. And the sheer scale of the preclinical success here is absolutely wild. In humanized mouse lymphoma models, they achieved persistent drug expression in the blood for over a year from one shot. That led to complete lymphoma regression and drastically improved survival rates. The fact that a single injection maintained therapeutic serum concentrations for over 365 days is absolutely astonishing. It's a total game changer for drug delivery. Now, when you compare this to other advanced therapies, specifically CAR-T, a really strong case starts to build. Don't get me wrong, CAR-T is an amazing cancer treatment, but it is a logistical nightmare. This AAV method, however, requires absolutely no patient-specific manufacturing, no extracting and engineering your T-cells outside of the body, and no grueling chemotherapy beforehand. It's framed as a significantly cheaper, highly scalable, off-the-shelf alternative to the incredible complexities of personalized cellular therapies. But hold on a second, we need to pause and talk about a critical flaw here. The major limitation of TransJoin is that it lacks a true off switch. If a patient suddenly develops severe toxicity or dangerous side effects, well, you can't just unplug the IV anymore. Once this continuous biological factory is turned on and churning out antibodies, you can't easily adjust the dose or just rapidly terminate the production. So how exactly do we fix that? Introducing the remote control upgrade, TransSkip. TransSkip is an inducible system where the therapeutic expression is actually turned on via a splice-switching morpholino. It elegantly solves that off-switch problem by giving doctors temporal, on-demand control over the drug's production inside the patient's body. It works like a brilliant biological chain reaction. First, the AAV vector is administered into the patient, but it sits there totally dormant. Second, when the doctor decides it's time for treatment, a specific, clinically established morpholino is administered. And third, this triggers a genetic process called exon skipping, which basically flips the switch and starts the continuous production of the therapeutic protein. This incredible mechanism gives doctors repeated on-demand control without having to keep the factory running 24/7. And the data? The data shows it works beautifully. TransSkip successfully achieved repeated induction of these therapeutic proteins for at least 36 weeks in the lab. This novel inducible gene expression system proved to be highly effective over incredibly long periods, all while maintaining its potent anti-tumor activity. So it gives you the drug exactly when you need it and only for as long as you actually need it.
But it is definitely time for a quick reality check. All the incredible efficacy data we've discussed so far currently relies entirely on mouse models and xenografts. There is absolutely zero human clinical data right now. We have to view this objectively. This is a highly innovative, deeply fascinating proof of concept, for sure, but it remains entirely preclinical.
Because we don't have human data yet, there's a massive list of unknown human immune responses. Like, how will a real human immune system react to the AAV vector? Will we develop neutralizing antibodies that just destroy it immediately? What about cytokine release syndrome, or CRS, or severe neurotoxicity known as ICANS? These are all major safety unknowns for continuous CD3 engaging therapies that we simply cannot predict perfectly from mice alone.
Furthermore, there are uninvestigated long-term safety issues to consider. What happens to the liver after years of being a dedicated drug factory? Is there a risk of insertional mutagenesis, where the vector accidentally disrupts normal DNA? What about chronic B-cell aplasia? To answer these critically important questions, scientists still desperately need to investigate these specific long-term toxicity risks in larger animal models, like non-human primates. We also saw a very specific scientific hurdle in the study with a variation they called oncoskip. The researchers tried a dual therapeutic concept here. They were hoping a KRAS-targeting morpholino would both activate the drug production and suppress endogenous KRAS proteins at the same time. Unfortunately, while the exon skipping worked perfectly to start the factory, the KRAS protein levels remained largely unchanged. It's just a gentle reminder, biology is incredibly complex, and these dual-action concepts still need some serious refinement. So, the crucial point to take away from all this is that despite these hurdles, the horizon here is incredibly bright. This platform isn't just a one-trick pony for cancer or blinatumomab mimics. Just look at these broad future applications. It's essentially a universal delivery truck that could theoretically manufacture other antibodies, cytokines, hormones, enzyme replacement proteins, and even CRISPR-Cas9 gene editing tools right inside your body. Which brings us to our final provocative thought for today. Will a single injection eventually replace the pharmacy? I mean, imagine a future where instead of picking up pills or sitting for hours under an IV drip, your medicine is manufactured directly from within, perfectly dosed, completely on demand. The transjoin and transskip platforms are just the very first steps, but they are a truly fascinating glimpse into the future of medicine. Thank you so much for joining me for this explainer. I hope it sparked your curiosity, and I will catch you next time.